So let us continue the chapter and we start with an experiment to show that CO2 is essential for photosynthesis. Now this is an important experiment. It can be asked in exam, maybe. So the experiment is we take two healthy potted plants having same size. We keep them in a dark room for 3 days. Now what we do next is we place each plant on a separate glass plates. In one, we place a watch glass containing potassium hydroxide with KOH. KOH is formula for potassium hydroxide. Now what is the function of KOH? KOH is used to absorb CO2. KOH absorbs the CO2. Okay, so in this plant, what will happen? CO2 is practically not there. KOH has absorbed CO2. So this plant does not have CO2. Okay, so logically, with the absence of CO2, this plant will not be able to take part or participate in photosynthesis. Now, this doesn't have KOH, so it has not absorbed CO2. CO2 is present. So this will be able to perform the process of photosynthesis. This will be able to perform the process of photosynthesis. Okay. So if this will be able to perform the process of photosynthesis, it will be able to make the food called as starch. So when we will check this plant for the presence of starch, it will show that there is starch and this plant has performed photosynthesis. Hence, it will be evidence that CO2 is required for taking part in photosynthesis. That's it. That's the experiment in nutshell. So how do we perform the starch test? We drop the iodine solution on the plant. It turns blue. That's it. That indicates the presence of starch. KOH is used to absorb CO2. Now cover both plants with separate bell jars. Using Vaseline, seal the bottom of the jars at the glass plates to make it airtight. Keep the plants in sunlight for about 2 hours. Test the leaves from both plants using iodine. So leaf of plant kept without KOH turns blue because it has taken part in photosynthesis. It indicates presence of starch. Plant with KOH does not turn blue, means it has not participated in photosynthesis, because with KOH means that the CO2 was absorbed. So this is the experiment in nutshell. This can be asked. If it is asked, this is the sequence by which students should be explaining this experiment. Let's move ahead in this chapter and see what is an autotrophic nutrition. This also we will try to understand by an experiment to show that sunlight is essential for photosynthesis. Previous experiment was explaining us that CO2 is essential. This will explain that sunlight is essential for photosynthesis. So keep a plant in dark room for 3 days to destarch the leaf. Destarch means all the starch will be used up, all the food will be used up. Cover a leaf partially with black paper. This is black paper. It has been covered partially using a black paper. Expose the plant to sunlight for 3 to 4 hours. Remove chlorophyll from the leaf and perform a starch test with iodine. Covered leaf part shows brown color. Exposed leaf turns blue due to the presence of starch. So what has happened? You covered this part. The remaining part has performed photosynthesis. It has made food for itself. The starch test is done on it. Iodine is put. It has turned blue. This part was not able to perform photosynthesis. It has not made food. It has remained brown. That's it. This experiment is used for determining that sunlight is essential for photosynthesis. Let's move ahead. Let's see what is autotrophic nutrition. Autotrophs also need other raw materials such as water and minerals like nitrogen, phosphorus, iron and magnesium. They are taken up from the soil. Where will it get these minerals? Nitrogen, phosphorus, iron and magnesium, they are taken up from the soil. Nitrogen is an essential element for protein synthesis. It is absorbed as inorganic nitrates or nitrites, or it is taken up as organic compounds prepared by bacteria from atmospheric nitrogen. Okay, now let's move ahead. Let us see what is a heterotrophic nutrition. It is the nutrition in which an organism depends on other living organisms for food. So it is not, autotroph is making its own food like plants. They are making their own food by the process of photosynthesis. Heterotrophic nutrition is a nutrition in which an organism is depending on other living organisms for food. Organisms take and use food by various ways. Example can be some organisms break down food outside the body and absorb it, like fungi, like bread, mold, yeast, and mushrooms. Some take in food and break down inside the body, like animals. Some organisms take food from organisms without killing them, that is parasite. Like the leech, which is in our hair. Sometimes we get leech in our hair. It's a parasite. So it is not killing the host, but it is determining its nutrition from the human body. Okay. Example of parasites is lice, ticks, leech, and tapeworms. Okay, so I hope it is understood. Some organisms are breaking down food outside the body and absorbing it, like fungi is doing to bread, molds, and yeast and mushroom do this similar thing. Some take in food and break down inside the body, like animal. It kills the deer, it eats its meat and breaks down the food inside the body. Okay. Now let's move ahead for heterotrophic nutrition. Unicellular organisms take food by their entire surface. As the complexity increases, specialized parts are formed to perform different functions. Now let us take the example of amoeba. This is important. It can be asked in exam. Amoeba being a unicellular organism, how it is taking food. Amoeba captures food using temporary finger-like extensions called the pseudopodia. This is a finger-like extension. It looks like the human finger. Hence it is called a finger-like extension, pseudopodia, of the cell surface. They fuse over the food particle, forming a food vacuole. In this, food breaks down into simpler ones and diffuse into cytoplasm. The undigested material, the material that is undigested, is removed to cell surface and thrown out. So this entire thing we have to understand, and we should be able to write this in exam if it is asked. Now let us see paramecium. Paramecium takes food in at a specific spot by the movement of cilia, which is a hair-like structure covering the cell. Okay, that's it. Paramecium takes food in at a specific spot by the movement of cilia. Okay, understood. Now let's move ahead. Nutrition in human beings. Now we have come to the part where we will discuss the nutrition in human beings. How is the food absorbed? How is the food taken inside? How is the food digested? Everything. So let us see how the nutrition is carried out in human beings. We have tongue, we have mouth, we have esophagus, we have a stomach, we have diaphragm, we have gallbladder, we have bile duct, we have a liver, pancreas, small intestine, large intestine, anus, and appendix. Now, when it comes to making diagrams, I always encourage students to make diagrams from the book called as Lakshmi Singh Manjeet Kaur. There is a very good science book, Lakshmi Singh Manjeet Kaur. That book needs to be referred. It has multiple good and beautiful diagrams. I recommend students to practice diagrams from the book Lakshmi Singh Manjeet Kaur, and they have to make the diagrams using that textbook, and the best diagrams will come out of it. So I encourage students to refer to the book Lakshmi Singh Manjeet Kaur for the diagram of the nutrition in human beings, like the digestion process. Now let's see nutrition in human being, food digestion. How does it start? It starts with the mouth, the breakdown of complex food with the help of enzymes into smaller absorbable molecules. So the food enters our mouth. The digestive system of the human body starts with the mouth. In mouth, food is chewed with the help of teeth and muscular tongue, and it is mixed with saliva, which is secreted by the salivary glands, which is secreted by the salivary glands. Teeth are used to crush the food into smaller particles. Saliva contains salivary amylase that breaks down starch to super sugar. This is an important line. People ask that what does saliva contains? We will say saliva contains salivary amylase, which is an enzyme that breaks down starch to simple sugar. Let's go ahead. There is another experiment now to show the action of saliva in food digestion. This is starch solution with saliva. If you put starch solution with saliva, we don't get blue color. If we put starch solution, if we put starch solution with saliva, we don't get blue color. If we put starch solution without saliva, we get a blue color. So experiment to show the action of saliva. Take 1 ml starch solution And in test tubes A and B, add 1 ml saliva to test tube A and leave both test tubes undisturbed for 20-30 minutes. Now add a few drops of dilute iodine solution to the test tubes. In test tube B, blue color will develop. It indicates presence of starch. In test tube A, no color changes occur because starch is digested by salivary amylase. So starch is there with saliva. So what has happened? Starch indication is the blue color. So if starch is there and saliva is there, what has happened? No color change because starch is digested by salivary amylase. So this experiment shows the action of saliva. There is no saliva, so starch remains with blue color. Here saliva is there. Saliva has, what has saliva done? It has digested the starch, so the blue color is gone. That's the concept, that's the experiment. Hope it is clear. Let us move ahead and understand the food digestion. Now we come to esophagus. Esophagus is the food pipe, the stomach, and we will understand what is peristalsis. The lining of alimentary canal is soft for smooth passage of food. The muscles in the lining contract rhythmically to push the food forward. It is called peristalsis. The muscles in the lining contract rhythmically to push the food forward. It is called peristalsis. It helps to process the food properly in each part. From the mouth, food is moved through esophagus, the food pipe, and reaches the stomach. Stomach is a large organ which expands when food enters it. Its muscular walls help in mixing the food. Its muscular walls help in mixing the food thoroughly with digestive juices. Now let's move ahead and see more about food digestion. Stomach wall contains gastric glands. So the stomach wall will be containing gastric glands. What will the gastric gland release? The gastric gland will release hydrochloric acid. The pepsin. What is a pepsin? Pepsin is a protein digesting enzyme. Pepsin is used to digest the protein. Pepsin is a protein digesting enzyme. And mucus. HCl, what is the function of HCl? HCl creates an acidic medium. HCl will create an acidic medium which facilitates the action of pepsin. So HCl creates an acidic medium which facilitates the action of pepsin. It also helps to destroy the harmful bacteria. Now the mucus protects the inner lining of the stomach from the action of acid. Sometimes HCl acts on the wall causing a burning sensation. This is called acidity. Okay, so everything is important in digestion. Understanding what is released by the gastric glands, what is the function of all that, what HCl is doing, what is the function of HCl. HCl is creating an acidic medium which is facilitating action of pepsin. It is also helping to destroy the harmful bacteria. What is the mucus doing? Mucus is protecting the inner lining of stomach from the action of the acid. And what happens? What is acidity? Sometimes HCl acts on the wall causing burning sensation. This is called acidity. Now let's go ahead and understand more about food digestion. We have a sphincter muscle which regulates the exit of food in smaller amounts from stomach into small intestine. From stomach the food goes to small intestine because of the action of sphincter muscle. Small intestine is longest and highly coiled part of alimentary canal. Its only name is small. It is the longest and it is highly coiled part of alimentary canal. This is the large intestine. This is the small intestine, this colon inside. Okay. So in animals, length of the small intestine differs based on the food they eat. Another question, herbivores need a longer small intestine. Herbivores will require a longer small intestine. The herbivores will have a longer small intestine for cellulose digestion, and meat is easier to digest. The meat that we eat is easier to digest. So carnivores like us have a shorter small intestine. Now let us move ahead. Small intestine is the site of complete digestion of carbohydrates, protein and fats. It receives the secretions of liver and pancreas. So the small intestine will also receive secretions from the liver and pancreas. So what is liver secreting? It is secreting bile juice. Bile juice emulsifies fat. It is the breakdown of large fat globules into small globules with the help of bile salts. It increases efficiency of enzyme action. It is similar to emulsifying action of soaps on dirt. And it makes the acidic food from stomach alkaline. So this is important. What is liver secreting and what is the function of those bile juice is important. We stop our second session at this point.